Okay, now we are going to continue to major types of fermenter used in industrial fermentation. Basically, there are several types of fermenter that has been used in the industry, but these are some of the common fermenter that commonly used in industrial fermentation. The first one is stir tank reactor or agitator to circulate the oxygen. The second one is bubble column, and the third one is airlift fermenter. Supply oxygen to the culture through intake valve in the bottom of the vessel. Okay, this is the schematic diagram that shows to you the stir tank bioreactor. So when we talk about stir tank, definitely this fermenter have a component called as a stirrer, or we call it as a agitator, or we also call it as a impeller. So the function of the stir or agitator or impeller is to ensure that the distribution of the nutrients inside the medium is distributed evenly, as well as to distribute the oxygen evenly throughout the tank. This is the schematic diagram for bubble column reactor. So the bubble column is the result of the gas that is introduced into the fermenter tank at the bottom of the tank. So this bubble column will ensure the circulation of the medium as well as the gas throughout the fermenter tank. And this one is airlift fermenter. Same as well as the column fermenter, the gas is introduced at the bottom part of the tank, and the gas will push out the medium inside the tank. And all those fermenter will ensure will ensure evenly distribution of the medium as well as the oxygen. The oxygen is really required by the microorganism during the beginning of the growth, because in order for them to grow, they need to have the oxygen first. After that, then the fermentation will take place. There are 5 factors that influencing the growth of microorganism in the fermenter. The first factor is aerobic. That means how much the oxygen that has been introduced inside the tank in the beginning phase of the growth of the microorganism. The second factor is the consistency of pH. Mainly, the optimum pH for the microorganism to grow is 7.0, which is neutral. But some of the microorganism does require different pH, and this pH is custom made to the specific type of microorganism to ensure the optimum growth, the optimum production of the metabolite. Hence, we need to provide the microorganism with the optimum pH as per requested by the specific strain. The third factor that is also a crucial factor is the temperature. As you already learned before, there are 3 types of microorganisms that have a different temperature for growth. The group of temperature are psychrophilic for cold temperature, mesophilic for room temperature, and thermophilic for high temperature microorganism. So depending on type of microorganism that we are using on that particular of time, hence we need to provide them with the specific temperature. The fourth factor is supply of nutrients for rapid growth. As I said repeatedly before, each microorganism will require a specific set of nutrients. We can give them a very general type of nutrient just to let them grow. However, in order for us to ensure the growth of the microorganism is at the optimum rate, Hence, we need to provide them with the specific type of nutrient as they decide to. And the last one is the anti-foaming agent to alleviate excess foaming. Mainly during the fermentation, industry or the manufacturer or producer will add on the anti-foaming agent inside the fermenter tank to avoid over bubbly formation from the agitation or from the distribution of the medium. Because if there is a lot of bubble or foam produced inside the fermenter tank, it will affect the rate of metabolite being produced, and also it will affect the quality of the metabolite that has been produced. So to overcome this kind of problem, anti-foaming agent will be added inside the tank. And one example of anti-foaming agent that commonly used is a oil, such as olive oil or any vegetable oil, to reduce the formation of foam. Okay, now we are looking into some major microbiome products that been produced by different kind of microorganisms. As you can see from this from the table, it's already listed out some of the different microbiome products together with the microorganism that are responsible to produce the product, such as ethanol from Saccharomyces cerevisiae. This is the common type of yeast, and another type of yeast that are quite uncommon is Cloveromyces fragilis. Enzymes such as Aspergillus, Bacillus, Muco, Trichoderma, amino acids such as Corynebacterium glutamicum, vitamin, polysaccharide from Xanthomonas, organic acids such as citric acid from Aspergillus niger. So these are some of the example of the microbiome compound that commonly produced and commonly applied in different kind of processing or industry, such as food industry, such as agricultural, such as pharmaceutical, and so on. These some of the another sample. Okay, now we are going to look on how of the some of the microbiome product being produced. For the first product is amino acid. As you know, amino acid is the monomer that used to build up the proteins. So amino acid will turn into peptide, and peptide turn into polypeptide, and polypeptide will turn into a protein. So amino acid is a crucial monomer molecule to build up a protein. So one example of amino acid that commonly produced in the industry called as the glutamic acid. In the common and in the commercial, I believe all of you already heard the name of Ajinomoto. So Ajinomoto is a brand name or is a company name that produces glutamic acid that commonly used as a flavor enhancer in food industry, or we call it as a MSG, monosodium glutamate. So glutamic acid is one kind of amino acid that responsible to generate the umami taste. Umami taste basically is a taste that will complement each of the tastes that we have on our tongue, on our taste bud. So our tongue can detect the taste of sweet, saltiness, sour, and bitter. So there are 4 different tastes that our tongue can detect. So umami taste that produced by monosodium glutamate or glutamic acid is the sensation of taste that we complement each of the tastes that our tongue can detect. So that's what we call as an umami, and commonly we only use a small amount of MSG into our food, so we can reduce the usage of salt into our food, because the combination of salt and also MSG will increase or elevate the taste of the food that we are preparing. Okay, now let's see how glutamic acid being produced. Basically, glutamic acid being produced by using a microorganism called Corynebacterium glutamicum. So the name of glutamic acid is taken from the name of the microorganism, Corynebacterium glutamicum. And glutamic acid is being produced from the starter molecule called as alpha ketoglutarate. If you still remember the metabolic pathway of aerobic respiration, after the glycolysis process that produces pyruvate, the pyruvate will undergo a tricarboxylic acid cycle, or we call as Krebs cycle, whereby the pyruvate will undergo a cycle of metabolic pathway, and one of the molecule or one of the metabolite that been produced called alpha ketoglutarate. So alpha ketoglutarate is undergo several processes by Corynebacterium glutamicum to transform the structure from alpha ketoglutarate into glutamate. So this big picture is showing to you how the amino acid is being produced from different kind of starter molecules, such as serine from 3-phosphoglycerate, alanine from pyruvate, alpha ketoglutarate will turn into glutamate, aspartate come from oxaloacetate, and so on. So the amino acid that being produced basically come from a different starter molecule. Okay, where does we can apply glutamic acid? There are several applications of glutamic acid. The first one, as I said to you just now, in the food industry to enhance the flavor inside the food, as well as to reduce the usage of salt. We also use glutamic acid in beverage also as a flavor enhancer. In cosmetic as a hair restorer for hair loss treatment and wrinkles such as preventing aging, and in agricultural animal feed as a nutritional supplement and feed additive to enhance nutrition. Okay, so the second product is vitamins. So the vitamins that commonly sold in the market are basically in the form of synthetic vitamin and also in the natural vitamins. So natural vitamin, basically you can obtain it from your daily food intake, such as vitamin C you can take from citrusy fruit, vitamin K you can take from vegetables, you can have vitamin E from some of the food such as fruits and so on. So what is vitamin? Vitamin is an essential micronutrient that are required in a trace amount only. It is an essential nutrient, but you only need to take it in very small amount. And some of the vitamin cannot be synthesized by mammal, such as us. Human is a mammal, but are essential for metabolism. So vitamin basically can be divided into several categories, such as fat-soluble, water-soluble, vitamin B complex, and vitamin-like substances. So to recall your knowledge in biochemistry, there are two types of vitamin. There are two categories of vitamin. The first one is fat soluble and second one is water soluble. For fat soluble is ADEK, ADEK. That means in order for the vitamin to be hydrolyzed, to be absorbed by the body, our body need to have a fat. So that's why people need to eat fat in their daily intake, because some of the vitamin does require fat to solubilize the vitamin of A, D, E, and K. Meanwhile, the rest of the vitamin falls under water soluble vitamins that only require water to dissolve the vitamin so that the body can absorb it easily. Okay, so these are some of the vitamin together with the microorganism that are responsible to produce it, such as vitamin C. We can get it from Corynebacterium species, biotin from fermentation of Serratia marcescens, multiple enzyme system of Bacillus sphaericus. We have vitamin B12 from the fermentation of Propionibacterium shermani, Pseudomonas dinifitricans, vitamin A from freshwater microalgae Euglena gracilis. Okay, this vitamin also produced by using fermentation. So as an example, vitamin B12 production, also known as a cobalamin, is a water-soluble vitamin, and some of the microorganisms that are commonly used in the production of B12, such as Streptomyces griseus, Streptomyces olivaceus, Bacillus megatherium, Bacillus coagulus, Pseudomonas dinitrificans, Propionibacterium fluodenrenchi, and Propionibacterium shermani. So from all of these example of microorganism, Propionibacterium fluodenrenchi and Propionibacterium shermani are the two common bacteria used in the production of vitamin B12 in industry. So as a schematic diagram, basically we need to have a specific media that been formulated according to the propionibacterium requirement, together with the pH and also some of the supplement, and then we are going to ferment it under anaerobic condition for 70 hours. After that, we are going to transfer the medium into another tank and undergo for aerobic fermentation with some addition of dimethylbenzene midazole 0.1%, and then we are going to withdraw and filtrate all the metabolite and purify the product until we got the cyanocobalamin with 80% purity before we can sold it into the market and use as an additive in the different industry. Okay, next we are going to look into organic acid. So organic acid basically is any acid that been produced during the fermentation of microorganisms such as citric acid, lactic acid, propionic acid, and so on. Different microorganisms or different bacteria will produce a different kind of organic acid together with different kind of sugar or different kind of carbon source. Organic acid produced through metabolism of carbohydrate, which is sugar. Organic acid accumulates in the broth of the fermenter, will undergo separation and purification processes, and some of the microorganisms that are commonly used during the production of organic acid, such as Aspergillus niger, Aspergillus clavatus, and Penicillium luteum. Okay, this is some of the schematic diagram that showing to you the processes involved in order for us to produce the organic acid. The first thing first, we need to have the pyruvate. As you already know, pyruvate will be produced whether in the aerobic condition or anaerobic condition. Is a product that comes from the breakdown of glucose, and the pyruvate will undergo a different metabolic pathway under anaerobic condition that will produce different kind of organic acid. As we can see right now, terminal end products such as pyruvate or alcohol will produce lactic acid or propionic acid. Incomplete oxidation of sugar, which is glucose, will produce citric acid, itaconic acid, gluconic acid, and dehydrogenation of alcohol with oxygen will produce acetic acid. Some of the microorganisms will produce a single type of acid, and some of the microorganisms will produce a mixture of organic acid with different percentage. Okay, where does the organic acid be applied? In this case, we are going to see the application of citric acid. So citric acid is widely used in food industry as a flavoring agent to give the sourness taste for your drinks or for your food. So if you have a food or drink that have the lemony or that have the sourish taste, more commonly the taste is come from the citric acid. Besides that, the citric acid also are responsible as a preservative to enhance, to prolong the shelf life of the food, and also act as antioxidant inside the food. In the chemical industry, citric acid also used as anti-foaming agent for treatment of textile and for metal industry as a chelating agent. And pharmaceutical industry, the usage of citric acid as a blood preservative, preservation of ointment and cosmetic as well as source of iron. Okay, the last metabolic product is enzyme production. So as you still remember, enzyme is an amino acid, is a protein that responsible to accelerate the chemical reaction in the cell. So some of the enzyme production, such as Taq polymerase enzyme, is widely used in polymerase chain reaction and other reaction. Cellulase obtained from E. coli used to degrade cellulose in plant cell. Okay, some of the enzyme also been used for daily life products such as detergent. Some of detergent they have an addition of lipase and protease to degrade the dirt that fat base and the dirt that protein base such as blood. So this enzyme will help to accelerate the breakdown of the stain and dirty from the clothes. Extracellular enzymes are secreted outside the cell. The enzyme makes recovery and purification process simpler compared to intracellular enzyme. Intracellular enzyme must be purified from thousands of different cell proteins and other compounds, and the microorganism-based enzymes are GRAS, or generally recognized as safe, GRAS, important status when it is used in food processing. So GRAS means that the microorganism used during the production is recognized as safe and wouldn't cause any harmful effect to the consumer, especially human. These are some of the applications of enzyme in industry, such as vinegar production. The enzymes are responsible in alcohol fermentation by yeast. So we are going to look more details about vinegar production in chapter 2. Textile industry, denim jeans treated with cellulose from Trichoderma raceae and Aspergillus niger to give the fading effect on and texture. If you can see, or if you do have the jean with the faded color scheme on your jeans, the faded color basically as an effect from the treatment by using enzyme. Detergent, as I said just now, protein from Bacillus subtilis is a component in laundry detergent to remove and degrade protein stain, and as well as in food and beverages such as pectinase, xylanase, and cellulase to improve the liberation of juice from pulp, mean the separation of the juice from the pulp to create a clear juice, such as apple juice. And pectinase and amylase also been used in juice clarification. Okay, thank you. So we are now we already completed our chapter 1. So I do hope that you can recall and that you can go through all the videos and all the material for the chapter 1 before we can proceed with our first assessment, which is quiz 1. Okay, thank you.